All right, so this is a lesson that gives you an overview of what photosynthesis is. Our learning objective for the lesson is to know the chemical formula of photosynthesis, to know the general structure of a leaf, and to know where glucose is produced and where they're stored inside a cell or inside the leaf. If we look at the metabolism definition that we had from last time, part of it is about providing energy for vital processes inside the cell, and so that the living thing can carry out all the function that it has to. Um, Photosynthesis is something that I believe you've learned in middle school, and the process is quite straightforward, and I trust that you know the input and the output of photosynthesis. Here's a diagram to assist you. So without looking it up on the internet, use just a diagram, come up with the word and chemical equation for photosynthesis. Assuming that you've tried it, I'm going to now move on and show you the answer. So photosynthesis has an input, which is carbon dioxide. You can see all the arrows going into the plant shows you that it is an input. So in a chemical equation, the input is on the left-hand side of the arrow, whereas the output is on the right-hand side of the arrow. Remember, the arrows in biology or in chemistry, if we're dealing with chemical reactions, it always mean the process of the reaction. So that's the reason why we've put the word photosynthesis. It's basically a process. Now, sunlight, although it is going into the plant, we don't really consider it as a direct input of photosynthesis. It's more like a source of energy rather than a matter that goes into the process. So it is an input, but it is a form of energy. Chlorophyll, we have put it underneath the arrow because it is not something that is being used up or processed on or involved. It is rather a place where the entire process happens in. And chlorophyll is a little machine or a pigment that helps to capture that sunlight. On the right hand side of the equation, we have glucose and oxygen. When we say the word chemical formula, we're referring to those that are written with the symbols. So 6CO2, 6O, H2O, C6H12O6, O, 6 oxygens, or 6O2. Now, this on the top, we call it chemical formula. If I ask for the word formula, then you'll be using the actual English word for it. Carbon dioxide plus water is going to produce glucose and oxygen. So don't mix up the terms. One's called a word equation, the one at the bottom, and one's called a chemical formula, which is on the top. Now, I do expect you to know the equation or the chemical formula for glucose at this stage. It's C6H12O6. You just have to memorize that, and it's not that difficult. All right, the next part is to try and get to know the structure of the leaf. Now, in your notes, you have this fill in the blanks activity. Use only the diagram to help you. I don't want you to search on the internet or search on Google or search AI for this. Use just your common sense and the diagram to help you fill in the blanks. Pause the video and do that now. Okay. So here's the answer. The cuticle layer is made up of wax, all right? And it is a layer that helps the plant to prevent dehydration. Now, I don't want you just to memorize that fact, but I want you to understand how. So we look at the material wax. Do you remember which biomolecule wax is under? Out of the four biomolecules, you have protein, you have carbohydrate, you have lipid, and you have nucleic acid. Then, after you found out what the wax belongs to, I want you to think about the characteristic of that category of biomolecules. And that's going to lead you to how wax prevent dehydration. So if you haven't done this lesson in class, pause the video, have a think about it, try and write something down, and then I'm going to give you the answer. All right, so wax is under the lipid category. Now, if you remember from Unit 1, there is a reason why I gave you all the chemistry basics in Unit 1. Lipid is a non-polar molecule, meaning that inside lipid, the way that the elements or the atoms are being arranged generates a molecule that doesn't have two opposite charged ends. So we call it non-polar. It doesn't have any partial charges on it. Now, if something is non-polar, that also means that it is hydrophobic. Fear water, hydrophobic. Now, something that is hydrophobic is not going to like water. It doesn't mix with water at all. So if you have a cuticle layer that is made up of a non-polar lipid layer, then that means water cannot go from outside of the leaf into the leaf through the surface. But at the same time, water inside the leaf cannot escape or evaporate to the surrounding environment through the waxy layer as well. So that is the reason why it helps to prevent dehydration. You're sealing the top of the leaf. Remember, the leaf is exposed to sunlight all the time. It's supposed to do that. It's designed to do that. The problem is if you're constantly exposed to sunlight, you're also at the risk of just evaporating all your water and become dehydrated. And we don't want that to happen, which is the reason why, by God's design, we have a waxy layer on top of a leaf. Now, that also means that it is kind of hard for gas, which is the carbon dioxide that the plants need to get into the leaf as well. So we're going to look at how they're doing it later. Okay, so let's move on from the cuticle. The largest proportion of the chloroplast, if you look here, it is actually located in a layer called the palisade. The palisade is a rather thick layer, and every cell in the palisade is jam-packed with chloroplast. Those are the little organelles that contains the chlorophyll, and they capture your sunlight for you. So the sunlight, in order to get to this palisade layer, it needs to pass through the cuticle. Thank God that it is clear or transparent. And then it needs to pass through the upper epidermis layer to reach the chloroplast, which is most of it is in the palisade. If you look at the upper epidermis and the lower epidermis layer, you probably won't see the little green organelles, which are the chloroplast, meaning that these two layers, the upper and the lower epidermis, are not involved in photosynthesis. Now, God is very efficient. If you don't need the material and you're not involved in the process, He won't put that material there. He will save it and leave it for places that actually does the job. There is very smart distribution of material. Okay, so most of the photosynthesis, as you can predict, looking at where the chloroplasts are, they happen in the palisade layer. And then we also have the spongy layer, which are circular, and there are a lot of space for gas to flow through to the palisade layer. They also have a tiny bit of chloroplast. You can see it here. So that means if not all the sunlight is captured by the palisade layer, then some of the sunlight is getting through. The spongy layer still has the ability to capture the rest of that sunlight and use it for photosynthesis. So a small amount of photosynthesis does happen in the spongy layer. Okay, so last two slides, we talked about the cuticle, how it prevents the loss of water, but then it creates a problem of not allowing gas to pass through. And that's the reason why we have these things called the stoma, or sometimes we call it stomata as well. It's just the singular and the plural form. So the stoma is a little opening that is controlled by the guard cells. And the guard cells can actually open and close the stoma to prevent the loss of water, but to allow the oxygen and the carbon dioxide to flow in and out. And these guard cells actually have sensory systems in them, and they respond to the temperature and the humidity of the surrounding environment. So if the humidity is low and then the temperature is pretty high, there is a high risk of dehydrating, then the stomata will be shut closed by the guard cells. Oppositely speaking, if the temperature is not too hot, there's a bit of sunlight, humidity is pretty high, you're not going to lose a lot of your water, then the guard cell is going to keep it open so that carbon dioxide can enter through the stoma and get to the palisade layer. Okay, after talking about the structure of the leaf, we're going to dive a little into the chloroplast. So as you can see, a chloroplast is a membrane-bound organelle, which is seen in eukaryotic cells. Only eukaryotic cells have these, if you remember from Unit 2. Now, the chloroplast structure, we're going to look at that in detail when we do our photosynthesis lesson in detail. All right. So the only thing that I want you to know for this lesson is the chlorophyll is located, the green pigment is located in the thylakoid, and they are responsible for capturing sunlight and use it for photosynthesis. Now, inside the chloroplast, photosynthesis are being divided into two major phases. You have the light reactions and the Calvin cycle. We also call it light phase and dark phase because light reactions that happens in part of the chloroplast, it requires sunlight to happen. And the input for that part is water, and the output is oxygen. Whereas the carbon dioxide is the input for the dark phase, the Calvin cycle. And as an output, we have carbohydrate, or you can call it sugar. Specifically speaking, it's glucose. So after producing the glucose, where does it really go? If we look at the structure of a plant cell, we have little organelles, which is again membrane bound. They're called the amyloplast. So the starch grain, you can call it. It contains the starch produced. So remember, chloroplast, they produce glucose. And from Unit 1, you know that glucose is a monomer. Now, monomers, if you want to store them, it's not the most efficient way of storing. So what you want to do instead is chain the monomers together. So chain the glucose together into a very long chain through chemical bonds and jam pack them in the form of starch. And in that case, you can actually pack more molecules of glucose in the form of starch into the same amount of space. So if you pack them as a glucose, you may only able to get 100 units into one amyloplast. But if you pack it in the form of starch, you might be able to get 150 molecules of those. Now, if we look at the picture on the left-hand side, it is actually stained amyloplast underneath the microscope. So what the scientists did was that they took a piece of leaf and they removed the chlorophyll, which is the green pigment that absorbs light. Because if you don't remove the chlorophyll, the colors kind of interact with each other and you can't see the amyloplast clearly stained with iodine. Iodine is a chemical that you learned in Unit 1 that can react with starch to generate a darkish bluish color. All right. So in order to stain and see if there are amyloplasts and if there's starch inside a leaf, you must first remove the chlorophyll. And this is exactly what we're going to do. The following class, we're going to test and see if the leaf have starch in it. Now, to do that, I don't really want to give you the method of how you can remove chlorophyll and how you can stain the leaf. I want you to figure it out. So in your notes, you have a hyperlink. If you click there, it's going to give you the structure of a leaf and a lot of statements, all right? So statements looking like these. It tells you what chemical or what process can do what. Now, I want you to figure out, number one, in order to remove all the chlorophyll, how do you remove the cuticle so that you can get inside the actual leaf and the plant cells? Number two, how do you break the cell wall and the membranes, the cell membranes, which are the phospholipid bilayers? Number three, how can you extract the chlorophyll and remove all the green pigment so that it's not going to interfere with the color that you're seeing after it's been stained? So use these statements to help you, and they should be ready by next class so that we can dive in and do our experiment. Okay, we're done for today.